Hello and welcome to Learning Made Easy, your destination for all things science and technology. Today, we're going to be talking about monoclonal antibodies, a powerful tool in modern medicine and research. Monoclonal antibodies, MABs, are a type of protein that are produced by a single type of immune cell, known as a B cell. They are used to identify and target specific antigens, such as bacteria, viruses, and cancer cells. These antibodies are produced by a technique called Hybridoma technology, which was first developed by Cesar Milstein and Georges Kohler in 1975. In this video, we will take a detailed look at the procedure for preparing monoclonal antibodies, as well as their various applications in medicine and research. First, let's understand the procedure of preparation of monoclonal antibodies step by step. 1. Immunization. The first step in preparing monoclonal antibodies is to immunize an animal, such as a mouse, with the antigen of interest. This is done by injecting the antigen into the animal's bloodstream, where it will stimulate the immune system to produce B cells that can recognize and bind to the antigen. 2. Isolation of B cells. Once the animal has been immunized, B cells that produce antibodies specific to the antigen are isolated from the animal's spleen or lymph nodes. 3. Fusion. The B cells are fused with a type of cell called a myeloma cell. Myeloma cells are immortal, meaning they can continue to divide and produce more cells without dying. The most common method of cell fusion is through the use of a chemical agent fusogen called polyethylene glycol, PEG. PEG works by creating a temporary pore in the cell membrane, allowing the two cells to physically merge together. Another method is through the use of electrical pulses to fuse the cells. When fused with B cells, these myeloma cells provide a long-term source of cells that can produce the same monoclonal antibody. 4. Selection. After the fusion, the resulting cells are grown in culture and screened for the ability to produce the desired monoclonal antibody. To ensure that only hybridomas producing the desired antibody are selected, a step called selection is performed. This is done by culturing the hybridomas in a medium containing a combination of drugs, such as hypoxanthine, aminopterin and thymidine, HAT medium. This medium contains aminopterin, which inhibits the activity of the enzyme dihydrofolate reductase, DHFR, a key enzyme in the production of thymidine. As a result, only those hybridomas that have the DHFR enzyme from the myeloma cell, and not the B cell, will survive. Additionally, thymidine kinase, TK, is also involved in this selection process. TK is an enzyme that helps to convert thymidine to thymidine monophosphate, and is present in B cells but not myeloma cells. If a hybridoma has the TK enzyme from the B cell, it will survive in the medium containing thymidine. The cells that produce the desired antibody are then selected and grown in large numbers. 5. Purification. The monoclonal antibody is then purified from the culture supernatant using techniques such as protein purification, chromatography, and centrifugation. Now that we understand how monoclonal antibodies are prepared, let's take a look at some of their applications. 1. Pregnancy test. Monoclonal antibodies are used in pregnancy tests to detect the presence of human chorionic gonadotropin, HCG, in a woman's urine. HCG is a hormone produced by the placenta during pregnancy. The test works by using monoclonal antibodies that specifically bind to HCG, and a positive result indicates pregnancy. 2. Cancer treatment. monoclonal antibodies are used as targeted therapies for cancer treatment, such as Herceptin for breast cancer and Rituxan for non-Hodgkin's lymphoma. 3. Immune disorders. Monoclonal antibodies are used to treat immune disorders such as rheumatoid arthritis and psoriasis. 4. Diagnostics. Monoclonal antibodies are widely used in diagnostic tests such as ELISA, Western blot, and immunofluorescence. 5. Transplantation. Monoclonal antibodies are used to prevent organ rejection in transplantation. 6. Infectious diseases. Monoclonal antibodies are used to treat infectious diseases such as respiratory syncytial virus, RSV, and hepatitis B. 7. Autoimmune diseases. Monoclonal antibodies are used to treat autoimmune diseases such as lupus and multiple sclerosis. 8. Cardiovascular disease. Monoclonal antibodies are used to treat cardiovascular diseases such as heart attacks. 9. Blood disorders. Monoclonal antibodies are used to treat blood disorders such as hemophilia and thrombocytopenia. 10. Respiratory diseases. Monoclonal antibodies are used to treat respiratory diseases such as asthma. 11. Gastrointestinal diseases. Monoclonal antibodies are used to treat gastrointestinal diseases such as Crohn's disease. These are just a few examples of the wide range of applications for monoclonal antibodies. They have revolutionized the field of medicine and continue to play an important role in the diagnosis and treatment of many diseases. I hope this video has helped you understand the concept of monoclonal antibodies and their various applications. If you have any questions, please leave them in the comments below. And don't forget to like, share, and subscribe to our channel for more informative videos on various scientific topics.